Hello, everyone. In this video, we're going to be looking at the cell, and for chapter 6, we're going to be looking at the differences between prokaryotic and eukaryotic cells, and we're going to be looking at um, the eukaryotic cell in more detail, including all of the organelles. Now, this chapter may be a review for many people because one of the things that is foundational to the study of biology is learning about the cell. So the cell is something that is taught to many students in their grade school education. So in this video, I'm going to give an abridged version of the lecture because there's no point in me just reading off the slides. I'm going to highlight some of the major and important parts of the cell. I'm not going to spend that much time on this lecture video. So let's get started. So this here is a paramecium. Paramecium is a great example. It's a very large eukaryotic cell, and you can see that in the inside of the paramecium, it's filled with all the organelles that are typical for a eukaryotic cell to function. So we're going to go over the different components of the cell, including how energy and matter works inside cells, the genetic information, how that's processed, and then interactions with the environment. So eukaryotic cells have internal membranes that compartmentalize their functions. So eukaryotic cells have these smaller functional units called organelles. Only eukaryotic cells have organelles. Now, the other domains of life, bacteria and archaea, are consistent of prokaryotic cells, meaning that they do not contain organelles. Now, the word prokaryotic means pro means before, and karyotic means nucleus. So, prokaryotic cells are the cells that evolved before the evolution of a nucleus. Eukaryotic, EU stands for true, and karyotic means nucleus. So eukaryotic cells are cells that are true nucleus cells. Um, so if you're not in the domain bacteria and archaea, if you're one of the kingdoms of life that are found within eukarya, then you are made of eukaryotic cells. Bacteria and archaea, though, are made of prokaryotic cells. There are some common components that are shared between all cells of life. All cells of life have a plasma membrane, so you must have a plasma membrane to form the structure of the cell. There's a fluid substance that is inside of those cells, and we call that the cytosol. The cytosol is where all of the components that do live within the cell are floating around in other words. Um, all cells have some chromosomes. So even though prokaryotic cells do not have a nucleus, they still have DNA, they still have chromosomes, and they carry their genetic code within that. So all cells have chromosomes. All cells have ribosomes. Ribosomes are the units that are responsible for creating proteins. So not only do eukaryotic cells need proteins, but prokaryotic cells need proteins as well. They need proteins to function. Proteins are responsible for our chemical reactions. So that's important. Prokaryotic cells are characterized by having no nucleus. They have a DNA in a region called the nucleoid. Basically, the DNA is clumped and it's usually generally isolated into a certain region. It's not surrounded by a nuclear envelope, so it doesn't have an actual nucleus. Prokaryotes do not have the subunits called organelles. They do not have any membrane bound um, organelles. And they have cytoplasm that is bound by the plasma membrane. The cytoplasm is the interior component of the cells. Now, here is a diagram of a prokaryotic cell. So, in the prokaryotic cells, you can see that you have a nucleoid, which is the region where the DNA is usually clumped together. And then you have ribosomes that are located within that. And so, depending on the bacteria, for example, um, while all cells have a plasma membrane, some cells can have a cell wall, some can have an additional wall structures, some can have what we call a glycocalyx. These are a thickened protective substance that will surround the bacteria cell. You'll also see that in this diagram that the cell has what we call fimbriae, which is a fancy word for cellular hairs, and flagella. These are used for movement. Okay. Um, and so this is the typical structure of a prokaryotic cell, and they are generally very simple like this and very small. Now, eukaryotic cells are a little bit more complex. They're characterized by having the DNA in a nucleus, and it's bound by what we call a double membrane, which we'll get to that in a minute. They have membrane bound organelles. They have subunits inside of themselves, like little factories that are able to do jobs. And they have a cytoplasm in the region between the plasma membrane and the nucleus. So eukaryotic cells are generally much larger than prokaryotic cells, almost like to a, in I would say average, like eukaryotic cells are about 100 times bigger than prokaryotic cells. And so, why is there a size limit to cells? If you guys think about it, we're not able to see cells with the naked eye. There are some cells that we can see with the naked eye. There are some structures that are created that are single-celled organisms. For example, eggs. So when you're looking at an egg, it's a single-celled organism. So the reason why cells are generally small is because there's a metabolic requirement that sets an upper limit on the size of the cells. And so the plasma membrane, the coating that surrounds the cell, is a selective barrier, which we're going to cover a whole lot in the next chapter. That it only allows certain things to go through. It only allows certain types of things to go through the membrane, and it only allows a certain amount to go in the membrane to service the requirements that are needed within the cell. So there is a surface area to volume ratio for cells. So as a cell increases in size, its volume will get proportionally as large, um, and it's usually more than its surface area. So as a cell increases in size, it is not able to do all of the things that it needs to do um, based on the surface area. You need more surface area to do all the interactions that are required to do all of the chemical reactions that are necessary within the cell. So in this diagram, you can see this is the structure of the plasma membrane. The plasma membrane is made up a lipid bilayer. So remember in chapter 5, we covered phospholipids. The lipid bilayer is made up, it's two layers of lipids with the hydrophilic heads facing towards the outside and the hydrophobic tails facing inwards. The plasma membrane is filled with proteins that are necessary to do all the chemical reactions and communications that are needed between cells. And so, once again, we're going to cover plasma membranes very much in depth in chapter 7. So here, this is just showing you that as you increase um, the surface, the volume, you are reducing the surface area that is necessary um, to for life, for the space that's required for all of these chemical reactions to occur within the plasma membrane. So if you stay smaller and you divide yourself up, you can maintain your surface area and still do all of the necessary requirements that it is for an organism to survive. So there's internal membranes that divide the cell into components, and so we like to call them organelles. The cell's compartments provide different local environments or different functions that can occur. Uh, and so the basic fabric of biological membranes, just like with the plasma membrane, is that it's a double layer of phospholipids and other lipids and protein components. Plants and animal cells have most of the same organelles, so there are some that are a little bit different between the two. So here in this image, you can see a typical animal cell and all of the organelles that make up that cell, from like the nucleus all the way to things like the cytoskeleton. So the plant cell contains all of these components. One of your jobs is you do need to know the parts of the animal cell. Same thing with the plant cell. So the first thing we're going to talk about in this video is we're going to be looking at the nucleus. The eukaryotic cell's genetic instructions are housed in the nucleus, and then the reading of those instructions are carried out by the ribosomes. So the nucleus contains most of the DNA in a eukaryotic cell. Surprise, surprise, not all DNA is located within the nucleus, which we'll come to that later on. And then ribosomes will use that information from the DNA to make proteins. Now, the nucleus is the brain, it's the information center within the cell. The nucleus contains most of the cell's genes, and it's usually the most conspicuous organelle. It's usually the largest organelle that is in the cell. And so when you are looking at cells, and you can, for example, if you can see cells and you can stain them, the nucleus is the first thing that pops out for many people. Now, the nucleus is surrounded by a double membrane called the nuclear envelope. It surrounds and protects the DNA, and it then separates the DNA of the cell from the outside components of the cell and from everything that's in the cytoplasm. The nuclear envelope is a double membrane, just like the membrane of the plasma membrane. Each membrane consists of a lipid bilayer. So here in this diagram, you have a nucleus that is attached to a rough ER. The rough ER is not considered part of the nucleus, but in the diagram, they want you to see and realize that the rough ER, the other members of what we call, you'll learn next, is the endomembrane system, is attached to the nucleus. But for the most part, the nucleus is its own separate individual entity. So the nucleus has several regions that we need to talk about. Um, this here is showing you that the nuclear envelope, is made up of a phospholipid bilayer. There's two layers there of phospholipids, which is a little bit different when you're talking about The plasma membrane, where the plasma membrane is just one layer of lipids, a bilayer. So the nuclear envelope is a like a two-layered system. So it's super protected. Inside of the nucleus, you have several regions. You have the overall nucleus itself, and then there's generally a region that is much darker and more not compact, but it is compact, and that is the nucleolus. The nucleolus is the region where ribosomes will be made. DNA will exist in the form called chromatin inside of the nucleus. Chromatin is the thread-like form of DNA. So when DNA is not undergoing replication, or if it's not, or if the cell's not undergoing replication, it it will be loose and free flowing in a chromatin form. It's like strand or strings. You can imagine it like that. And so that we call that form the chromatin form. Now, the nucleus is filled with pores called nuclear pores. And these nuclear pores, they allow for only certain items to go in and out of the nucleus. One of those being ribosomes. So ribosomes are manufactured on the inside of the nucleus, but then they are sent outside of the nucleus to do its job inside of the cell. So ribosomes can go through nuclear pores, and so can RNA, messenger RNA that needs to travel outside of the nucleus to go and be read by ribosomes to create proteins. Now, pores line the structure, and we call pores will line the nuclear envelope, and we call those a pore complex, and it regulates the entry and exit of those molecules. There is evidence for a nuclear matrix, a framework of protein fibers throughout the interior of the nucleus. And basically, these are scaffoldings that are in place to make sure that the nucleus doesn't collapse in on itself. Now, ribosomes, what are ribosomes? I mentioned earlier that they are like these little factories. They're these units that are created that allow for messenger RNA to be read. They are complexes made of what we call ribosomal RNA and protein. So ribosomes build proteins in generally two different locations within the cell, in the cytosol, and so we call those free ribosomes. Those are the ribosomes that can travel anywhere in the cell, anywhere that they're needed to do some sort of chemical reaction. So ribosomes will travel and it will search for messenger RNA, and it will read the messenger RNA, which would then allow for the creation of proteins. Another location for ribosomes is on the outside of the endoplasmic reticulum, or what we call the nuclear envelope. And so this is where ribosomes will be bound to the endoplasmic reticulum, or what we commonly call the rough ER. So in this image, I just want to show you that ribosomes are they're actually made up of two subunits. One's a larger subunit, one a smaller subunit, and they come together. They land, it's like they form a sandwich with messenger RNA, and it's read to produce a protein. From the image here, there's a TEM image showing you the endoplasmic reticulum and how it's lined with these dark spots, which represent ribosomes. In the next video, what we're going to do is we're going to cover the endomembrane system, which is going to include things like the ER, the Golgi, lysosomes, and other uh, parts of the system.